In this video, we are going to discuss about the enzyme. First of all, we know about what is metabolism. So, the sum of all chemical reaction which are takes place in the body of living organisms is called metabolism. We can also define metabolism, the set of biochemical reaction that occur in living organism in order to maintain life is called metabolism. The term metabolism is derived from Greek word meaning change. The concept of metabolism was first of all given by Ibn Nafis, who stated that the body and its part always undergoing change. There are two types of metabolism, or we can say that metabolism can be divided into two types, catabolism and anabolism. Catabolism, which is also known as a destructive process, are the process in which large complex molecules are broken down into small and simpler molecules by the release of energy in the form of ATP is called catabolism. For example, respiration, in which the glucose molecule react with oxygen to produce carbon dioxide and water with the release of energy in the form of ATP. And sucrose, when combined with water, it is converted into glucose and fructose with the release of energy in the form of ATP. So this process is known as catabolism. Anabolism, it is the opposite process of catabolism and can be defined the process in which small or simple molecule combine to form large and complex molecule by using the energy is called anabolism. Their example is photosynthesis. Carbon dioxide and water, both of them, they are small and simpler molecule in the presence of sunlight, enzymes, and chlorophyll. It is converted into glucose with the release of oxygen and water. Again, glucose and fructose, these are small and simpler molecule. They combine with one another in the form of, in the presence of ATP and converted into sucrose and water is produced. So the characteristics of all living things such as breathing, reproduction, growth, and response to stimuli are the outcome of biochemical reaction takes place inside the body of a living organism. All these reactions are controlled by a specific protein called enzyme. Enzymes word is derived from two Greek words. In mean inside, enzyme mean yeast and can be defined. The enzymes are biological catalysts which increase the rate of chemical reaction in living cell without being used in the process is known as enzyme. Or enzymes are biological catalysts which speed up a chemical reaction without alter itself. The term enzyme for the first time is used by German scientist William Coney in 1878. And approximately 4,500 different types of enzymes are discovered in different types of living organisms. Now we will go to explain the characteristics of enzymes. Enzymes are biochemical catalysts and possesses the following important characteristics. Nature. All enzymes are protein and are secreted by cells. Enzyme act like a catalyst. It only speed the rate of biochemical reaction without being used in the process. Enzymes are specific in action. They are not specific only for substrate but also for the kind of biochemical reaction. Enzymes are required in very small amount as compared to the amount of substrate. Then what is substrate? The molecule that fits the active site is called its substrate. Substrate can be defined. The substrate the substance upon which an enzyme acts is called substrate. Specific active centers. Enzymes have specific active center called active site, which is attached to the substrate. We can define active site. An enzyme molecule has on its surface a region called an active site. We can also define active site. In each enzyme molecule, there is present a particular site where the substrate binds. This site is called active site. Enzymes are very sensitive to the changing of temperature, pH, etc. They may stop their activity. The enzyme can be divided into two types. Intracellular enzyme, these are the enzymes which are produced and perform their function within the cell. Such enzyme is known as intracellular enzyme. For example, mitochondrial enzyme. Extracellular enzymes are those enzymes which are secreted outside the cell and perform their function. And their example is lipase, amylase, pepsin, etc. Enzymes are not consumed during the biochemical reaction and can be used again and again, just like a biological. That's why it is known as biological catalyst, just like a catalyst. Enzyme lower the activation energy. Activation energy are those energy which is responsible to start a reaction, to start a process. Such energy is known as activation energy. So enzymes do not initiate the reaction but increase the rate of reaction by lowering the activation energy. Enzyme have the property that it's lower the activation energy. When the activation energy is lower, the reaction can be started, which is required for substrate. Some substance interact with enzyme to prevent it from working. It is called enzyme inhibitor. For example, poisons, drugs, etc. Some enzyme work properly when a cofactor is attached. So then a question is arise, what is a cofactor? So cofactors can be defined. It is atoms, group of atoms and molecule that join the enzymes, changing their shape and making them functional. It is known as cofactor. Again, we repeated the definition of cofactor. Cofactors are atoms, group of atoms and molecule that join with enzyme, changing their shape and making them functional. It is known as cofactor. So cofactor is a non-protein organic molecule compound that is required for enzyme as a catalyst, and it can be considered helper molecule for enzyme. The main type of cofactor is prosthetic group, coenzyme and activator. Then what is prosthetic group? If the cofactor is a non-protein like a metallic ion, for example zinc, copper, iron, then it is known as prosthetic groups. Then what is coenzymes? Some cofactors are small organic molecules called coenzymes. Like enzyme, they are not permanently changed in the reaction. And many of the coenzymes are derived from vitamins and minerals that are essential for life. And the absence of these cofactors can lead to vitamin and mineral deficiency diseases. For example, lecum, vitamin B, produces beriberi. Example of coenzymes are NAD plus, FAD plus, and NADP plus. Specificity of enzymes. Enzymes are generally very specific in their action. They are specific not only for substrate but also for the kind of reaction. Every enzyme, due to its special chemical structure, can recognize a particular substrate. Enzyme that catalyze one reaction may not catalyze another. For example, catalase is the name of enzyme which only catalyze the decomposition of hydrogen peroxide. So catalase react with hydrogen peroxide and convert it into hydrogen and oxygen gas. Protease is the name of enzyme which convert protein into amino acids. Amylase is the name of enzyme which work only for the digestion of starch. Lipase is the enzyme which convert lipid into fatty acid and glycerol. So the specificity of different enzymes is determined by the shape of their actioside. The actioside possesses specific geometric shape that fit with the specific substrate. For example, this is the substrate and these are the enzyme and these are the actiosides, while the remaining structure of the enzyme is known as the framework. So the actioside easily fit within the substrate and convert the substrate into products. What is activation energy? The energy required to initiate a chemical reaction is called activation energy. So all chemical reaction required activation energy. Enzyme basically lower activation energy. When chemical reaction takes place, some amount of energy is required to break chemical bond in reactant substrate. Such energy is called activation energy. Enzyme lower the amount of activation energy, thus in the presence of enzyme, reaction proceed at a faster rate. Enzyme act in many ways to lower the activation energy. For example, they change the shape of substrate molecule or bring them in correct orientation. For the reaction of hydrogen peroxide, which is converted into hydrogen and oxygen gas, the activation energy is 86 kJ per mole is required without catalyst. and just 1 kilogram per mole of energy is required. 1 kilojoule per mole energy is required in the presence of an enzyme. If, for example, an enzyme is not available, then hydrogen peroxide is converted into hydrogen oxygen gas and it requires 86 kilojoule per mole of energy. But when enzyme is present, only 1 kilojoule per mole of energy is required to convert hydrogen peroxide into hydrogen and oxygen gas. So that's in this simple reaction we can conclude it that enzyme basically it lower the activation of energy. You can see here in the diagram that A plus B, these are the reactant, will be converted into products. So if the activation energy is lower down, for example, if the enzyme is present, then the curve is very less. But when enzyme is absent, so it will pass through this line and convert it into products, and it will take a large number of time. So 86 kilojoule per mole of energy is required, and this process takes place 2 hours, or we can say many time. When enzyme is available, it will take very less time, and within a second, within a less time, the hydrogen peroxide is converted into hydrogen in a oxygen gas. Mechanism of enzyme action. Mechanism means step-by-step chemical reaction. There is a small portion in enzyme that is involved in catalysis, which is known as a actiosite. It is the form of depression or pocket on the surface of enzyme molecule. When enzyme attach with the substrate to form enzyme substrate complex. You know that enzyme when it react with substrate, enzyme substrate complex is produced. The actiosite of enzyme catalyze the reaction into products. The enzyme substrate complex is converted into enzyme products, and then enzyme is released and product is released. The enzyme substrate complex break and the enzyme and the products are released. In reaction form, enzyme substrate complex, then enzyme product complex, then enzyme as well as products are released. So here there are two models which explain mechanism of enzyme action. One is known as lock and key model and the other is known as Kochland inducement hypothesis. So the lock and key model, this model was presented by a German scientist Emil Fischer in 1894. According to this model, enzyme act as a lock and substrate act as a key. As one specific key can open only a specific lock. In the same manner, the specific enzyme can transfer only specific substrate into products. The enzyme must have the correct geometric shape to fit the substrate. According to this model, the actiosite is a rigid structure. There is no modification in the actiosite before, during, or after the reaction. This is the substrate, this is the enzymes. They combine with one another to form the enzyme substrate complex, then enzyme product complex, then products as well as the enzymes are released from one another. If you like the video, please share, subscribe, and watch. Thank you.